Now is recording. Okay, guys. Good evening. My name is Sandro Renato Dias. I'm professor from CEFET-MG, working at the computing department, and I'm working with a lot of areas in computer networks, distributed systems, programming, and bioinformatics. My undergraduation was in computer science with master's degree in electrical engineer and prototyping field in computing engineer. And my PhD was in bioinformatics, and this is the theme of this workshop. So we're here with the Professor Estefanía from Salamanca University, a huge researcher and professor there. I had a pleasure to meet her in Portugal, Bragança, when I was there as an inviting professor in the Instituto Politécnico de Bragança. So Professor Estefanía will start with the presentation and will present herself too. And after that, we will talk about bioinformatics in a practical way. We'll use some tools to understand some issues in bioinformatics about pollen in proteins. Okay, so thank you, Professor Estefanía, again for this participation, and
thank you to you too, because it's a pleasure to have the opportunity to be with you all today. I'm sorry if I have to ask for a little bit of comprehension, just in case I fall asleep, because here in Portugal is quite late, but I'll try to do my best. So just in case you see my eyes are closing, just say something, aloud. Okay, as Professor Sandro...
I'll call you.
Okay. As Professor Sandro told you before, my name is Estefanía Sanchéz Reyes. I'm a researcher at the University of Salamanca, and I'm working at the moment in the palynology group. That's my line of expertise. I've been working in this science since during the last 15 years or so. So I will have a lot of information to give to you, but I just want to give you some global ideas in order to a better comprehension of the practical exercises that Professor Sandro is going to show to you afterwards. So just let me share a short presentation I've prepared. Give me a second. Okay. Can you see the presentation, Sandro?
Yes.
Yes. Okay. Okay. I have put the first slide full of different pollen grains, because maybe you have the idea that pollen grains are all the same, but that's not true, and we are going to have the opportunity to know a little bit more about them during this workshop. So let's start with this, I hope that very interesting workshop entitled Tasting Bioinformatics with Pollen and Proteins. As I told you before, I'm a palynologist, and this is the science that focus on the study of pollen grains and fungal spores. In fact, the main topic is normally, or it has been in the past, pollen grains, but now it is becoming more interesting to work with fungal spores, since they have they are involved in the allergic processes, as we will see afterwards. As you know, the pollen grains are the male part of the plants that is going to be in charge of the fecundation of the plants. The pollen transport to the stigma of a flower can be taken by different vectors. One of them is the wind, and this is the most important one considering the allergy processes, because these pollen grains, the anemophilus pollen grains, are the main allergic ones. However, there are also other vectors that can be responsible for the transportation of these pollen grains. I think that we all are used to talk about bees, but there are other animals in the case of zoophilus pollen grains. For example, bats or even mammals, even human beings, we can be the vector of this kind of transport. And in the end, also the water can be a vector in which pollen grains can be transported from one plant to another one. Here you have just a very a slide a little bit full with different images, figures, considering the morphology of pollen grains. But pollen grains have different features that allow us to identify and to recognize the different pollen types. Here you have, I don't know, can you see my mouse moving around the screen? Yeah, okay. You have here, for example, I always like to show this pollen grain to my students because I think it's very, and also in this picture it's even more clear that it's similar to the ears of Mickey Mouse. You see here, this is a pollen grain of Pinus, genus Pinus. But we have, for example, here Acacia species. They all have this genera, all have the species belonging to this genera. They all have this pollen grain that is very similar to a football ball. Or also you have here the crocus pollen grain, very similar to a tennis ball. Or you have here different pollen grains belonging to other plants. Well, the idea is that they are going to be quite different one from another ones. So with the proper training, people are going to be able to identify all of them, because this work of identification is being doing manually. Okay, until now there have not been machines or devices doing this type of work. Here you have some real pictures of different pollen grains in order to show you how different they can be. So you have, they're beautiful. When I started to do this kind of research, I totally fell in love with them, because it's just like when you are having a look under the microscope, you never know which pollen grains is going to appear the next. So it's quite a mystery sometimes. So, well, here you have some pictures, and some of them belongs to the more allergenic pollen grains, and we will see them with more detail afterwards. With fungal spores, because I told you that palynology is the science that is in charge of study pollen grains, but also fungal spores. With fungal spores, it's more or less the same. They are going to have also different features, which allow us to identify and to differentiate them one from another ones. Here you have also different shapes. I'm not going to give you names of fungal spores, because we are going to focus on this workshop on pollen grains. And once these researches are carried out, we can apply the results, or we can apply this type of researches to different sciences. One of them is the melizopalynology. And in the end, what we normally do in this kind of research is to identify and count the pollen grains that we can find in honeys. So in the end, we are going to be able to determine the geographical and botanical origin of this pollen, of these honeys. That's quite important in the market, in this market, because we are going to be able to identify frauds, for example. And this is a very interesting research. I'm not going to give you more details, but if you are interested on knowing a little bit more, you can contact me afterwards. I'm going to give you my email and my contact, just in case you are interested in knowing a little bit more about all of this. We can also use pollen grains as another taxonomy feature in plant taxonomy. It can be used also as other feature more, as for example the size of the plant or the number of leaves per branches or so. Or also in paleopalynology, we are going to be able to explain the distribution of some species or groups, or even to reconstruct the plant landscapes that were in the past on Earth. That's quite interesting, and it also contributed in the past to the improvement of the Pangea theory. So that was really, really interesting in the past. And another application that the study of pollen grains and fungal spores is in forensic palynology. This was in 1959 was the first time that the policemen used these studies in order to solve a crime. There was a couple that were traveling on a trip along the Danube River, and one of them disappeared during this trip. So the policemen tried to identify different clues around this crime. So they gave the boots of the other person to Wilhelm Klaus, that was a palynologist around in the area, and he was able to reconstruct the vegetation, the specific part of the Danube River coast on which this person had been given a walk, for example, and they were able to identify the specific zone considering this vegetation, so they could found the corpse, and they could found this person guilty of killing. I don't know what happened because I've lost the presentation. Can you see the presentation now? Here, okay.
Yes, we can see.
Okay, sorry, it was my fault. So imagine it has been also used, for example, in analyzing the holy sheet in order to establish the date of this holy sheet, or also in studies in relation with mummies, in order to know which were the processes that they were normally carrying out in order to elaborate these mummies. But also another application is in agronomy, because we can use this kind of research in order to make crops prediction or to prevent and treat the fungal pests. So we'll be able to develop phytosanitary treatment schedules in a better way. But the main application is in medicine. Why? Because the pollen grains, or at least anemophilus, most of the anemophilus pollen grains are considered allergenic. Okay, I'm not going to give you a lot of information about the process, but they are able to activate type 1 immune response. I think that we are all more or less familiar with all this kind of sciences processes because of or due to the COVID-19 that we are suffering at the moment. So this is the science, the palynology science in relation to allergy processes, and in order to monitor, for example, the airborne concentrations in different cities and locations, is called aeropalynology. And this is the line of expertise I'm working at the moment, and we also wanted to show to you some information related to that and related to bioinformatics as well. I suppose that you are familiar with these kinds of news, because every single spring they all appear in every single country. Every single year is going to be worse than the year before, and that's because the pollen concentrations in air are going to be really, really high. This year is not different, and as we already know, it's been a very, very difficult year to allergy people. We don't still know if it's because of the COVID situation, or it's because maybe the vegetation this year is higher, it's in a great number because of the meteorological conditions, or what. We are studying that at the moment, but in the end, it's been a very difficult year for these people. And because of the importance of all these relations to allergy, you can see here a worldwide map in which you can find all the pollen and fungal spores monitoring stations. And I'll invite you to go to this link, because you will see that, for example, in Europe, there are a lot of monitoring stations. But even though Brazil is a very big country, you just have a few ones. So it will be very interesting if we be able to increment this number. And I'll tell you that we are working on that at the moment. Here is the device we normally use for this kind of research. It's a very small device that reproduces the normal breathing of a healthy lung in normal conditions. That's a flow rate of 10 liters per minute of air. Okay, in the end, we are going to, I'm not going to give you more information about that, but we are going to be able to identify the different pollen grains that are appearing in this device along the year. So we'll be able to establish a pollen calendar like the one you have here, in which we can see which pollen types are appearing in January, February, March, April, May, June. That's really, really useful for medical doctors in order to give advice and to adjust the treatments for the allergic people. There were a couple even more in the past. We saw that there was some differences in the researches, because we were able to identify non-pollen grains in very specific days when there were storms, very strong storms. There were no pollen grains in the air. These devices were not able to capture any pollen grain, but there were a lot of people that went to the emergency services with asthma processes and, very ill. So we started to use another device, another different device that instead of capture pollen grains, it is able to capture proteins. So we saw that in these very specific stormy days, there were no pollen grains, because the storm broke all the pollen grains, liberating all the proteins into the air. So we were not able to see the pollen grains, but the proteins that in fact are the allergic causes of these allergic processes were in the air. So related to that, I'm going to show you just a little bit of these 4 pollen grains that are the main ones in terms of allergenicity. The first one is the pollen grains that have all the grasses. is a rounded pollen grain with just one spot, one aperture, and it's very, very allergenic. I would say that this is the first cause of allergic process worldwide, but especially in, for example, in Mediterranean countries such as Spain or Portugal. Here you have which are the main proteins responsible for these allergic processes. As you can see, they have the three first letters of the first name of the species, the first letter of the second name of the species, and then a number that is related to the position in which these different proteins were discovered for the first time. Okay. The second one that was shown in the slide is the one related to betula. That's very important pollen grains in terms of allergenicity in the north of Europe, in the Euro-Siberian countries. And here you have the main protein related to allergy processes related to this pollen grain. It's Betula verrucosa. There is another one that is the main cause of allergenic processes in winter. We are studying that pollen grain at the moment here in Bragança, in Portugal, in the city I met Sandro, Professor Sandro. And it's provoking a lot of problems because it's a normal ornamental element used in parks and gardens and also in the cemeteries. There are other species, other genus that are normally in nature. They are not cultivated, but we can find that one as well in the city centers. Here you have the pollen grains. The name is Cupressus sempervirens. And here you have the main protein related to this, to the allergic processes in terms of this pollen grain, CUP-S1. And last, we have the oliveira, the olive tree. You know that this is this is one of the main crops in the Mediterranean area. So we can find these trees in, for example, in Portugal, in the south of Portugal, Spain, Italy, and it's causing a lot of problems to the people that are working on this in these crops. The name of this pollen grain is Olea europaea. That's the name of the species as well. And here you have the main proteins that were identified as responsible for the allergic processes, OLE1 and OLE9. Specifically, we are going to work afterwards with OLE1 with Professor Sandro and Bioinformatics. This is all I wanted to show to you because, I mean, there is a lot of information and I didn't want to give you all to you in just this 20-25 minutes, but just one more idea. We are working at the moment in terms of the COVID pandemic because some colleagues have demonstrated that there is a relation between the levels of pollen grains in the air and the infection rates by COVID. At the moment, they have only studied airborne pollen concentrations, but they are considering to study not pollen grain concentrations, but proteins coming from pollen grains in the air. So you will see that there could be a lot of applications of the bioinformatics that Professor Sandro is going to show to you afterwards. Here you have my contact. I'm working in this building at the University of Salamanca at the Palynology and Plant Conservation Research Group. Here you have my telephone number, just in case you want to call me, or my email address in case you want to make a consult or you have any doubt. So thank you very much for your attention, Professor Sandro. The floor is yours in this case.
Okay, thank you, Professor Estefanía. Please put your email in the chat for the students.
Yes.
Okay, guys. I'm listening my voice. I think it's your microphone, please, Estefanía. Okay. Yes. Now it's better. I don't like so much my voice, so hear me talking and hear me again. Okay, guys, let me share my screen to you. Okay, Estefania put her email in the chat if you want to contact her. Now, I think you can see my screen. I will put you guys in the other monitor so I can see you and me. Okay, let's start. What is bioinformatics? Bioinformatics is, I like to say, that is another language that connects biology and computer science. But bioinformatics is not just computer science, but a lot of other areas like engineer, medicine, biology, statistics, maths, physics, all of them working to solve biological problems. So Estefania said about the Olea quê? Olea pali, please.
Europaea.
Ah, sim, Olea europaea. Olea europaea. This is a plant, Stefania. What is Olea europaea?
That's the oliveira.
Oliveira, so it's a plant.
Yes,
and here
tree.
we have a tree. Okay, sorry. I don't know nothing about biology. Well, this is the PDB, is a protein data bank. A protein, like Stefania said, is a piece of everything. And in this database, protein data bank, we have a lot of information about proteins. For instance, let me search here Olea europaea in PDB. We have here 178,747 structures of proteins here in this database. Well, Olea europaea, I don't find nothing, but if I search OLE1, this structure, for example, we have this information. This is a protein in this database, and what we have in this database is not this file. I'd like to show you. Sorry. Okay. I have to open the protein. Sorry, guys. If I click in this protein, I have all information about this protein. This is ligustrum. I forgot the name. Ligustrum what, Stefania?
Vulgare.
Ligustrum, yes, here. Ligustrum vulgare is the name of the organism that we can find this protein. And in this database, we have two formats of files, a PDB file and a FASTA file. This is a way to find some information about the protein. And in this file, okay, we have all of the amino acid residues of this protein. Each letter of this is an amino acid. And you can see them here in the, let me come back to the, okay, here. This is the visualization, 3D visualization of the protein. And all information about the how can I say that, the resolution of the structure of the protein and the method that was used to that, X-ray diffraction. So they used a huge equipment to use an X-ray in a synchrotron lab. They have the X-rays exiting in a curve. So when they cap that X-ray, they put it on a crystal of a protein, so they can solve the structure of the protein. And this structure is like this. It's opening. Okay. These letters are the amino acid residues of the protein. This is a cartoon way to see the protein, but we can see it in another way. For instance, in ball stick. Cartoon. Okay. Ball stick. If you see here, each ball of this is an atom, and the sticks are the connection between the atoms. Look here, we have, oh, where are you? Here, let me see, here. An aromatic ring here in the mouse isn't working. Okay, now you can see here an aromatic ring here, and the blue ball is nitrogen, the green here and the red. Here we have a lot of structures, but the green and the red are carbons, and the yellow here we have sulfur atom, and the red in the outside here are oxygen. And this connection, the connection between both, the interaction between both atoms. So we can see here the structure of the protein, atom per atom, that we can find in this PDB file. This is the FASTA file, and this is the PDB file. In the end of the file, here, we have the atoms coordinators, yes. So we have here the nitrogen, the residue of amino acid, the chain. So we got a lot of chains in this protein. I think they have 3 or 4 proteins, 3 or 4 chains. And the number of the residue, the number of the atom, and the X, Y, and Z coordinators of this atom, and all atoms of this protein. And this first residue of amino acid here is the same of this, is this letter, E. So this is a part of the protein. And the information that we have here above is all the information like this. That is the journal where the paper was published about this protein, about the resolution of this protein. Information about the equipment used to do the solution, the resolution of the protein, and some details of that, like the experimental details, the date that was done, the temperature, the pH of the solution, and the number of crystals used to solve this protein, and another information, a lot of information here. Okay, so why I'm showing you this? This file is used to publish this information of the protein, the structure, and all textual information that we have in the page before here. Okay, and what can I do with this? I can use this information to search about this protein, to study this protein, and to compare this protein with another one. Estefanía said that we have a lot of pollen grains from a specific family that can cause allergies. Okay, so we have here this specifically protein, and we have another databases like the UniProt. UniProt is another database that you can use to search too. We'll use this in a few minutes. And in this database, we have the FASTA file that is the same of this, that have just the letters of each amino acids of the protein. And we can use it to do an alignment between them. We can compare a protein with another one using the structure of the protein, but considering just the position of the amino acids in the line of the protein. In all of the structure, we can call this the primary structure of the protein. Okay, so let's start again with you guys. If you want to follow me, you need to open the rcsb.org. rcsb.org. I'll put this in the chat for you. Please open the site, and then we'll put in this field ole e uno. Are you doing this? It's okay. Yes? Okay, so, and here we have a lot of options, and we want to search for a structure, not just keywords to search, but a structure, specific structure, and we have this structure of the inflammatory response of to the OLE1 protein. So let's choose this. Yes. Yes. So the PDB will open this page. It's okay, everyone is in this page. Estefanía, are you there? Okay. Let me see. Yes, great. Thank you, Pedro, for answer. Okay, if you click in the name of the protein, this structure. Great, Julia, thank you. So we can see all the information of the protein. Okay, is everyone in this page now? The page of the protein, 6YOA. This is a PDB code. Is a code that used for all proteins in PDB. Each one will have another name for this, another code for this. Each of the almost 108,000 structures. So each one will have a code different by this. Okay, so here we can see the structure. Let's see the 3D view of the structure. Each protein have a different way to fold. A fold is this structure, this way the protein fold by itself. When we put try to think in a line. You put a line in the water, and this line will fold in a specific way. This is how the protein works. But each protein will fold in just a specific way. We cannot decide or try to guess how it works. Some programs try to predict a structure of a protein using just the FASTA file, the letter of the amino acids, the sequence of the amino acids of the protein, trying to guess this structure. But it's very, very difficult to guess. And the structure of a protein is directly related to his function. So this protein have this configuration, this conformation. And if we show this protein in a different way, we can see, okay. Using ball stick, we can see the sequence of atoms of this protein, like I showed before. And in this structure, we have here the beta sheet is this structure. The alpha helix is this structure, because we have an helix here. And the loop region is this structure here. So the amino acids have a conformation in the three-dimensional space here, forming just a loop, just a connection between the alpha helix and the beta sheets here. Beta sheets because we have a lot of structure in the same direction, like a sheet, a sheet of paper. And the alpha helix is because we have this configuration of the atoms. If we see the atoms, is more difficult to understand it because we have a lot of balls and sticks, so it's difficult to try to guess this structure, this cartoon structure. Okay, and here we can compare this protein to another one. So let's try to use another database. Okay, here, the UniProt. Open this one, please. And here we'll search specifically OLE-E12. UniProt. Here we have also a lot of information about proteins. This is another database, but they don't have the three-dimensional structure like the PDB. And the searching here. Wait a little. Have you got the answer there? I'm waiting, I'm still waiting here. But yes, let me come back what I got before, this. So I think you got this information.
It's not working for me too.
not working. Let me try again.
It's taking a lot of time for me as well.
Play one.
And I'm not getting the information we saw before. I don't know why. I don't know what's the problem. It has to be something with the web page.
Is searching, and not answering? Or you got a page?
I've got a page, but it's not. I can.
I'm trying. Oh, I think yes. Now I have here. This page. You got this page?
Yeah.
Okay. So we can click in the first one, major pollen allergen, OLE1, organism Olea europaea. So you can click in this code or another link. P19963 is the code of this UniProt database. And here also we have information about the protein and where the protein is found in the cell, and a lot of other information is here. But we will not search all details of this protein, but you try to find the FASTA. I use it just to show you another database of protein and to find the FASTA file. So click here in Format, FASTA. And we got this sequence of letters too. Like in the previous one, that we got this, this sequence. So we have two different proteins here, and we'll compare the both. This we find in UniProt. Yes. Let me see. Organisms Olea europaea, and in this organism is Ligustrum vulgare. So we have two different proteins from two different organisms. So we have to, we will compare the both. Okay, so we will use another, where is it here? No, we use another site, BLAST. I will send the link to you in the chat too. Are you following me? Are you lost? Is everything okay? No following? Yes, lost? Yes. Sleeping, trying to sleeping, not sleeping. Everything is fine. Okay, thank you, Guilherme. So, you got the BLAST site? Okay. I opened two databases and now a third site. BLAST is an alignment tool. Is a software that we use to align sequence of nucleotides or amino acids to compare DNA, RNA, or sequence of proteins. So we use the, oh sorry, I clicked there. So we'll use the protein BLAST. We'll compare a protein to a protein. If we use the nucleotide BLAST, we'll compare nucleotide to nucleotide and compare DNA with DNA. We can compare translated nucleotide to protein. So we can compare a DNA with a protein to search where that protein comes from. So which part of DNA can produce that protein. And the opposite too, a protein to a translated nucleotide. So we can search both sides. But now we will search protein to protein. So click here, protein blast. Okay. Protein blast. And now we need the both sequence. So I will come back here to the PDB of the Ligustrum vulgare protein, and I'll click here in Display Files, FASTA sequence. They show me all of these letters. I will copy all of them, put in my BLAST sequence. This sequence. BLAST sequence, chook. Okay. The default interface is this. So we need to, after paste the sequence here, we need to click here and align two or more sequences, because we'll compare two sequences specifically. Click here. I will not explain every configuration of this. The software will spend a lot of time with this. Just this exercise. And we'll come back to the UniProt to do the same. So we open Format, FASTA. Let me put the sequence here for you. The first sequence is in the chat too. Yeah, I'm sending to you. The first one is this, and the second one okay. I send both sequences to you, and the second one I will paste here. So the first one, the second one, and we will not change anything. So we'll click here in BLAST. And now we can drink a coffee. This is so fast because we have just two proteins. If we use the BLAST to search for between all proteins in the database, it can spend a lot of time doing this. So it's better to download the software in the database, to put in a server, and let your protein being searched in another proteins. We can use a whole protein or a small piece of a protein to search if we have another protein with that sequence specifically. Okay, so what we got here? A lot of information to BLAST of two sequence, the identification of the sequence, and here we have the information and the alignment of the proteins. If I click here in this first result, you got this page? Everyone got this page? Funny, yes. So if you click here, we can see the sequence of both. If you see in the chat, the first one starts with EDV and the second one EDY. No Y, I. So we have here EDI and EDV. So in this alignment, we can have the information of the difference of both. So we have this V and I here different, and this Y and H here. Here we have a P and a T. And nothing. Yeah, they have the same size, so we not have a place here where is missing some residue from the first or the second one. So both have the same size, and they are so close, so close. We have 127 amino acids identically from one to another one. So we can guess that these two proteins are quite similar, or they have the same function. And Estefanía can say to us why they are similar.
The thing is that people that is allergic to Oliveira, to Olea loparea, they are also allergic. They normally are also allergic to this other species, Ligustrum vulgare, because of this protein. They share the same, or more or less the same structure. So people that are allergic to Olea europaea, they're going to have clinical symptoms for a longer period due to other plant species that share the same proteins that they are sensitized to. So this is a huge problem for allergic people, because in the end they are suffering more than they are thought to be.
So
yes.
Both proteins can give the same symptoms of the allergies.
Yeah. Pedro is asking if this plus symbol in the middle line mark the differences between both sequences. I guess yes.
Yes, yes. And we can have a protein in the second sequence here that have more or less residues of amino acids here. And we can see a sequence between them with some marks here too. Yes?
You have to think that these kind of researches were the first ones that give the clue of the cross-reactivity of different pollen grains, and also why some people are allergic to, for example, a very specific pollen grain, but also to some fruits or some food. This is all because the proteins share the structure and composition in amino acids. So in this, in terms of knowing a little bit more about this, bioinformatics, it's, I would say, essential for knowing all these processes. So you can see here how biology needs the tool of bioinformatics for getting results and conclusions in most cases.
If we don't have the bioinformatics to search these and get these results, how can we guess if a person is will have symptoms of allergy with these two proteins? How can we test this?
A skin prick test. That's what we can do.
Put the protein in the skin to see if they have symptoms.
Yeah, they're going in, the medical doctors are going to pinch you some drops of the different proteins, and they are going to see if you produce a reaction. So depending on the proteins you are allergic to, you are going to get like a spot that it's, in case it is bigger than 3 millimeters, 3 centimeters, sorry, it means that you're allergic to that specific protein. So you can be tested first for the whole family, for example, and then in case you react positively, you can they can go further and analyze to which specific plant you are allergic to.
Okay. That's a huge contribution of the bioinformatics. So we don't need to do these tests, these skin tests, with all these proteins. And another way to search here is maintain the same sequence, the first sequence. Click on the align two or more. So we will search this sequence in the whole database. Don't click anymore, just this to reduce the sequence to just one, and you click in the blast. And what you got with this is the alignment of this sequence with all the database. So we can find another proteins that are similar to this one and that can cause the same allergies because they have the similar structure. So this will got so much time than the first one. And the blast can be used to compare protein to protein, DNA to DNA, so we can compare which part of our DNA is similar to the Drosophila melanogaster, that is the fruit flying. Yes. Okay, so here we have the Ligustrum vulgare and the Olea europaea,
and you have all the plant belonging to the same family. For example, Syringa vulgaris is another species, and Fraxinus excelsior is another species belonging to the same family. So people that is allergic to Olea europaea, they are going to have reactions to these other plants as well.
Yeah, and a few days ago, I knew the Syringa covidaris in my arm. Okay, we have the Fraxinus excelsior too. What is the Fraxinus excelsior?
That's another tree that normally that normally grow up really near to river banks. And these ones, the Fraxinus ones, normally pollinate in early spring, Oliveira in late spring, and Ligustrum in early summer. So if you are allergic to Ole E1 protein, probably you will have allergic symptoms since during the whole spring and also at the beginning of the summer. And because normally people that are allergic to Olea europaea, they probably are, because of these cross reactions, they are normally allergic as well to grass pollen. They will be suffering from allergic symptoms during the whole spring and the whole summer. So it's interesting to know all this in order to give advice and to take the proper prevention measures.
So we have all this information, and you can see there are a lot of bioinformatics tools that you can use. And here I do not have this alignment. They are processing, so you can see a multiple alignment here using this specifically tool. Trying to. Oh, you got a failure here. Request failed. Okay, try to do it again. Let me see here. Here we have each alignment with the first sequence with all of them. The first sequence is this that we put in the query field. And in the other link I clicked before, you could see all the proteins aligned like this, but in a multiple alignment. but I think it's not working now. Maybe because it's a lot of information. Okay, but you can see, you can try it again later, and you can see here they have 100 sequences they are trying to visualize here. And you can see in this page all of each alignment of sequence. We can do this with the FASTA file, searching just the amino acid sequence, or we can use another tool like the LSQ KAB is a software that we use in the Linux, the command prompt, to compare the 3D structure of a protein. Only by comparing this data, a doctor would be able to say if a person is allergic to some protein without the skin test. Estefanía, is there?
I mean, you need to know first that a person is allergic to a specific protein. And the only way to know that is by a skin prick test or immunoallergic testing. But after that, with these tools, with these bioinformatic tools, you can guess if being allergic to one protein could be responsible for being allergic to another one. And knowing which is the composition on proteins of the different foods or I don't know, pollen grains or other animals, for example, acres and all the possible allergenic particles, the doctor would be able to establish this cross-reactivity in this specific patient. But firstly, you need to know that this person is allergic to one specific protein. This information is not going to be given by the bioinformatic tool. You need to do the skin prick testing. I don't know if I answer your question.
Yes, you did. Thank you.
Great.
I see that with bioinformatics, you always know when the game starts, but you never know when it finishes because you can do a lot of things. I mean, you can just play with the bioinformatics tools and get even more information that the one you were supposed to get at the beginning. So that's quite interesting in my line of research.
Yes, we can get a lot of information of the proteins, comparing them, the structure, trying to guess where this protein came from, like I said. We can use the bioinformatic tools to do a docking. We have a lot of tools for this. Docking is I don't remember a software to do a docking. A docking is a software that we can use to compare a structure. Docking protein online. A structure of a protein and a pharmacus. How can I say pharmacus? Yeah, the pharmacus. Doctor. And
treatments.
We can see if this pharmacus will. Sorry. Excuse me. Say it again. I didn't listen to it.
I was just saying the treatments, the different treatments.
Yes. This we have a protein. In this we have the not the treatment, but the medicine. Yes, the molecule of the medicine. We can use this to try to find a better place where this medicine will act in this protein. So we can use a tool, a bioinformatic tool, to do that. So what this tool will do? They will run the protein in several ways and change the place of the medicine to fit in the protein, in a better way to fit the protein, so we can guess if this medicine will have a huge actuation in the brain, the person, the blood, whatever. So when you get a medicine, when you get a medicine with the information of new formula or better actuation, more success than the previous one, is because they changed a lot of bit in the pharmacos, in the molecule of the medicine, so they can fit better in the protein where this pharmacos will act in the organism. So
I guess this is being used as well for COVID.
Yes.
Okay.
Yes. Includes let me see. I don't know. I don't have a software here. They use it in COVID to compare the medicines for COVID with the protein of COVID. So they use this to try to guess if that medicine will act better or not act in COVID protein. So it's a way to use the bioinformatics. Okay, guys, any question? I think we talked a lot about a lot of things, and I try to show you tools of bioinformatics just for you to taste a little bit and to try to understand more, to learn more about bioinformatics. So you can send message to us, to Estefanía and to me. I put my email here too. Yeah, it's too much data. Yeah. I know. But in bioinformatics, we work with big data also. In my PhD thesis, I constructed a database with a billion of files of protein interactions to create a database where I can search for a protein structure specifically to try to find some interaction that can fit in that structure. So my thesis is to search an interaction to suggest to the researcher a new mutation in that protein to become that protein more stable and strong in very different situations like huge temperatures, high temperatures, or low temperatures. I don't know, different from the stability, the normal stability of the protein. So the objective is to gain stability in the protein, adding some mutations that can bring another interaction between the atoms of the protein, changing the residue of the amino acid of the protein. So is another way to search in bioinformatics, to research in bioinformatics with proteins, with DNA, with virus, with bacteria, and a lot of things we can do in bioinformatics. Okay, Estefanía, you want to say something more?
Nothing, just thank you for your assistance today, for your attendance. It's been a pleasure to be with you, and I hope that you have learned a little bit about what we normally do, Professor Sandro and me, in our research. And every doubt or it's going to be welcome, so just write to us, and we will try to answer your questions.
I sent to you the link for the what is this? It's a presence list for you to generate the certificate of this participation. So don't forget to fill it, please, this form. Oh, thank you, Mariana. Welcome to to research with us if you want to.
Maybe one day.
Yeah, it will be our pleasure. Okay, thank you, Otávio. So, if you guys want to ask something, we are here to answer you.
I have a question.
Yes?
To Professor Estefanía about what you said in the beginning of the presentation. How can climate change affect the pollen concentrations? You said this year has been difficult towards people with allergies. Can it be related to the climate change aspect of the situation?
Of course, for sure. We're studying that because we have seen that the flowering periods are becoming different in every single species. In some cases, they are becoming longer. For example, they need more time to get the complete biological cycle. And in other cases, it's the other way around. For example, in the case of some species, the pollination period is getting shorter, but becoming more intense. So for allergic people, it's becoming worse. That's the reason. Maybe it could be the reason. Might be the reason for this year to be so difficult compared to other years, because the meteorological conditions this year are quite warmer than years before. And also the rain, for example, can also influence on pollen concentrations. And as you may know, we are registering less rainfalls in the last years. So at least the meteorological processes are becoming, more extreme. So we have hotter summers and, for example, colder winters, and this is affecting plant species in all their biological cycles, which include, of course, the pollination periods. So definitely, I mean, it's been studied at the moment.
All right, thank you. Makes sense.
Thank you, Aline.
Okay. So, no more question? Any other question? Don't forget to fill the form.
And say that we were great.
Yeah. Excuse me.
I won't say just that they need to fill the form, but saying that we were great. If you are not going to say that, don't fill the form, okay?
Yes. Please. Okay. Thank you, guys. Thank you, Aline. Thank you very much. So you can finish now. Don't forget to fill the form to get the certificate of this participation. And thank you again. I will finish this recording.